Okay, lab 3, we'll talk about aseptic technique, cell culture handling, and growth curve through this lab. So first of all, what is aseptic technique? We talked about it before, but it is a technique that will prevent contamination on your culture or molecules. Okay, so by implementing this technique, you will prevent contamination of your materials that you're handling. So those materials will be pure cell culture, so culture made out of single cells only, and they should not be contaminated, of course, with other cells of different species. And if you are dealing with molecules that are isolated pure, like DNA, RNA, protein molecules, of course they should not be contaminated with other molecules of different sources. So this technique is very important, especially in the molecular biology lab. And this technique also prevents infection or exposure of operator by, implementing this technique. It'll also protect you guys from possible infection or exposure to dangerous chemicals. So what are the important things then in your aseptic technique? First of all, you have to have a clean workspace. So if your space is dirty, of course, you will have contamination more. So your workspace should be clean. So what workspace should you use? Mostly either clean room environment. So we don't have a clean room in the lab, but instead we have a biosafety hood that will generate very clean environment, clean workspace. Okay, and the workspace should be also disinfected or sterilized. Sterilized is not possible, but disinfection should take place. Disinfection means what? It's cleaning up using disinfectants, the chemicals that can kill microbials or living contaminants that can infect your materials. Okay, so the working area, or even if you're using tools or materials, then those should be sterilized. Okay, so those are important practices. And then sterilization, if you're dealing with maybe pure solutions, so growth medium, those should be sterilized. So understand the difference between disinfection and sterilization. Disinfection is kind of reducing the amount of infectants by using chemicals, by using different methods. But sterilization is what? Complete removal, complete killing of contaminants. So, for example, autoclaving, putting your solution into a hot chamber with pressurized steam in it, that'll eliminate, remove all the living cells. Filtration is another effective way. You can filter out possible contaminants using the filter that has very small pores. And then handling of behavior is very important. So the operators, especially you guys, when you deal with your clean materials, you have to minimize the exposure of those materials to the outside air, outside possible contaminants, by minimizing the exposure time. Okay, so don't allow any sterile materials to touch other things that are not sterile. Of course, those are the basic rule. And then working with the cells. So when you deal with the cells, when you grow them in the aseptic conditions, you may grow them on a solid media, like a petri dish plates, or liquid medium, like a growth medium made out of liquid. Okay, there are pros and cons of using different medium. If it's a solid medium, normally they come in a petri dish container. They are based on the agar, so they are solid, although they are kind of soft. Okay, liquid medium is made out of liquids, so they are contained in a maybe test tube, a flask, or bioreactor, fermenter. Okay. The main purpose of using solid medium is you can separate cells into a single if you use solid medium, so by maybe streaking. Okay, so if you get a clump of cells on a maybe streak or maybe a sterile loop, you can make a lot of streaks on your plate, and then rotate your plate, go through another streak, another streak, and another streak. Then by the end of this fourth streak area, you will dilute out the cells there are a lot here, and they are slowly diluted out. You can dilute out all the way to the single cells. So these are the single cells. Okay, so if you seal up this plate and grow them overnight, tomorrow you will come back and you will see those colonies. Okay? Actually, we call them colony forming units or colonies. Those are the clones, meaning they start out from a single cell and they grew into maybe millions of cells collected together there. So that's a colony or colony forming units. So you can isolate a clump of cells into single cells by sticking on a solid growth medium. Okay, and cell culture quantification through serial deletion. So if you have a liquid cell culture and you want to identify how many cells you have in the liquid culture, there are various ways of doing that, cell quantification method. One of the most accurate method is by doing a serial deletion. Okay, so if you have very concentrated cell within 1 mil of that cell culture, you may have billions and billions of cells. You cannot even count those cells, right? Then how can you make them to be countable? Okay, so if you can dilute that out, so maybe 1 mil of very concentrated cell culture, maybe into maybe a diluted cell culture, maybe that's a growth medium only, that's maybe 9 mils of growth medium. You take 1 mil out of this culture and mix it together. So 1 mil mixed up together, it's giving you 10 mil. So 1 to 10 dilution, right? And from there, you can do another dilution. There's a 9 mil of medium. You transfer from 1 mil here to here and mix it together, getting a 10 mil there, right? And you keep transferring there. So by the time you go to there, so that's going to be 10 to the minus 1, minus 2, 3, 4, 5, 6. You can dilute it all the way up to 10 to the minus 6 dilution there. Okay, so by the time you get there, you have about 1 millionth of dilution there. Okay, so if you transfer these cells to a plate, okay, and spread them, maybe transfer 1 mil or maybe 100 microliter, whatever the volume, transfer them and spread them on the plate. Okay, all of them will be transferred to a new plate. Okay, and then grow them overnight. Next day when you come in, from some of these plates, you will be getting the cells in the countable numbers. Countable means it's less than 200 colonies per plate. That's countable. Okay, this may be lawn of cells. It's all connected. There's no way you can count. Same way, they may be countable, but maybe up to make 200. Okay, then if you have like 17, that's definitely countable. Okay, 2 maybe too few. So if you get countable number of cells, just count number of cells. So 17 cells from there, and that was from what, 10 to minus 4. The original concentration will be what, 10 to the 4 times more concentrated. So you multiply 17 times 10 to the 4th there, then that'll be the concentration of original culture. Okay, so if you want to use that, so 159 colonies times 10 to the 3, so 159 times 10 to the 3, so that'll be what? So if you move 2 decimal points, then 1.59 times 10 to the 5 cells per mil of the concentration you found from there. Okay, so that's how you can quantify it. Yes, through serial dilution. That's one way of doing that. Another way of doing that is okay, so before that, so there's exercise 1 is on that method, serial dilution method. So we are giving you an overnight E. coli culture that's in the liquid medium. Okay, so I want you to make serial dilutions, so involving about 7 dilutions. Okay, so if you make serial dilution, so take 10 microliter of the overnight culture, and if you mix that with 990 microliter of liquid medium, so it's 10 over, if you combine them two, is what? 1000, right? 10 over 1000, that's a 1 to 100 dilution, right? So you make that maybe several times, so that's a 10 to the minus 2, and do it the same way another one is 10 to the minus 4, do it another one 10 to the minus 6, 10 to the minus 8. You can make about 4 of those dilutions all the way up to 10 to the minus 8 dilution, right? And you plate those onto a medium, the petri dish medium. Take 100 microliter of each to each medium and then spread them all and then grow them overnight. Can you count the CFU's colony forming units? Okay, and then estimate the concentration of the original culture. Okay, so this is in your practice batch record, so practice that using that. Okay, you can find that from your core shell. Okay, so that's exercise number 1. We'll get to do it face to face. I'll explain more when you get to do it. Exercise number 2 is a cell quantification method using a spectrophotometry. So in the lecture we talked about spectrophotometer when you determine the DNA double strand versus a single stranded form. Okay, that same machine can be used in cell quantification. Okay, so spectrophotometer has a light source. Okay, and then you place your sample in a cuvette special holder and you take the absorption. Okay, so it is known that if you are dealing with E. coli cells, bacterial cells, okay, and if the bacterial cells are dissolved in its liquid medium, okay, and then if you shine the light, the visible light of a 600 nanometer wavelength light, and if you your cell culture gives you 1 OD, optical density unit, okay, that is normally about these many cells per 1 mil, so 8 times 10 to the 8th number of cells. Okay. So if that's the case, then what if you get 0.5 OD? Then that'll be half of that, right? So that's 4 times 10 to the 8th cells. So that is a reference point you can use in your spec reading. If you're dealing with maybe eukaryotic cell that's much larger, like unicellular eukaryotic cell like yeast, and that OD reading means a little different. So 1 OD at 600 nanometer, in each cell it's only 3 times 10 to the 7th cells. Per mil. The difference comes from the size of the cell. E. coli cells are much smaller than yeast cells. Yeast cells may be about, I don't know, maybe 20 times larger. That's the difference. Okay, so spectrophotometry method is very quick and kind of be dirty method, easy and quick method to do it because you don't have to wait overnight to be able to quantify it. You can do it right away, right? So you can use this method. That will be the exercise 2, monitoring the cell growth pattern using the liquid culture. So we have the batch record in your core shell. You'll follow that. So you will be given it the overnight culture. You'll inoculate it into the cell culture medium, TXYT medium last week in the lab 2, and then you will observe the observance change through about 3-hour time period. And from the observance, you will convert that to a cell density, and based on that, you'll plot a growth curve. Growth curve means what? So if you have time versus number of cells, so using this equation, you'll be able to figure out the number of cells, okay, cells per mil. So if you do that, you will see that the cell growth will give kind of S-shaped curve. Okay, when cells are introduced into new medium, they kind of stay kind of put. They don't double right away. They don't replicate right away. But after a certain time, if they get adapted to new environment, meaning adaptation meaning cell physiology is changed. In a kind of new environment, they don't utilize the nutrients and all those things right away. So it takes time to get adapted and to get their maximum physiology by expressing needed genes and things. It takes some time. Okay, after they have enough enzymes and all those things made, then they start to double, going through the cell division cycle. So lag phase initially, and then log phase is the exponential growth phase where cells continuously double. And then eventually they will run out of their space and nutrients, and they stop doubling, and they just stay there, put. That's a stationary phase, okay? And then eventually they will kill each other because they are starved to death. So there will be a death phase. Okay, so try to observe that using your, spectrophotometer readings that you will get through the batch record. Okay, so that is what you need to do, to plotting the growth curve. Okay, so that is the lab 3. So you will be able to get the batch record downloaded from the core shell. Okay, that's it.